Hello everyone, we are here to discuss about subject Biopharmaceutics and Pharmacokinetics presented by Amit Varati, Pooja Patole, Karthik Shirke, Suyash Patil, Sanika Patil, guided by Mr. A.N. Dhage sir. Question: define drug absorption. Discuss physicochemical factor influencing GI absorption of drug. Definition: drug absorption is defined as process of movement of unchanged drug from site of administration to the systemic circulation. Physiochemical factors influencing absorption of drug. First, drug solubility and dissolution rate. Second, particle size and effective surface area. Third, polymorphism and amorphism. 4. Pseudo-polymorphism. Fifth, salt form of drug. Sixth, lipophilicity of drug. 7. pK of drug and gastrointestinal pH. 8. Drug solubility. First point, drug solubility and dissolution rate. First, dissolution is the rate determining step for hydrophobic and poorly aqueous soluble drug. For example, Griseofulvin and spironolactone. Second, permeation is the rate determining step for hydrophilic and high aqueous soluble drugs. For example, Cromolyn, sodium or neomycin. Third, prerequisite for the absorption of drug is that it must be present in the aqueous solution, and this is depends on drug aqueous solubility and its dissolution rate. Second point, particle size and effective surface area. Particle size may play a major role in drug absorption. Second, dissolution rate of solid particles is proportional to surface area. Third, smaller particle size, greater surface area, then higher will be dissolution is thought to take place at the surface area of the solute. Fourth, particle size reduction has been used to increase the absorption of large number of poorly soluble drugs. Fifth, for example, bis-hydroxycoumarin, digoxin and Griseofulvin. Two types of surface area. First, absolute surface area. Second, effective surface area. Third point, polymorphism and amorphism. Polymorphism. Depending upon the internal structure, a solid can exist either crystalline or amorphous form. When a substance exists in more than one crystalline form, the different forms are designated as polymorphs and the phenomenon as polymorphism. The polymorphism differs from each other with respect to their physical properties such as solubility, melting point, density, hardness, and compression characteristics. Thus, this change in physical properties and hence the absorption. Amorphism. Some drugs can exist in amorphous form. Such drug represents the highest energy state. They have greater aqueous solubility than the crystalline forms because the energy required to transfer a molecule from the crystal lattice is greater than that required for no crystalline (amorphous form). Fourth point, pseudo-polymorphism. When the solvent molecule entrapped in the crystalline structure of the polymorph known as polymorphism. Solvents. The stoichiometric type of adduct where the solvent molecule are incorporated in the crystal lattice of the solid called as the solvents and the trapped solvent of crystallization. Hydrates. When the solvents in association with drug is water, the solvent is known as hydrate. Hydrates or solvents are pseudopolymorphs where hydrates are less soluble and solvents are more soluble and thus affect the absorption accordingly. Fifth point, salt form of drug. While considering the salt form of drug, pH of the diffusion layer is important, not the pH of the bulk of the solution. Example of salt-weak acid. It increases the pH of the diffusion layer, which promotes the solubility and dissolution of the weak acid, and absorption is bound to be rapid. Other approach to enhance the solubility and absorption rate of certain drugs is the formation of salt formation, that is increases in pH microenvironment of drug by incorporating of a buffering agent, for example aspirin and penicillin. But sometimes more soluble salt from of drug may result in poor absorption. For example, sodium salt of phenobarbital, its tablet swelling and did not get disintegrate, thus dissolve slowly and results in poor absorption. 6 point pH partition hypothesis. This theory states that the drug compound of molecular weight more than 100, which are primarily transported across the biomembrane by passive diffusion. The process of absorption is governed by first point, the dissociation constant pKa of the drug, second, the lipid solubility of the unionized drug, third, the pH at the absorption site. 8 point drug stability. A drug for use may destabilize either during its shelf life or in the GIT. Two major stability problems resulting in poor bioavailability of the orally administered drug are degradation of the drug into inactive form and interaction with one or more different components either of the dosage form or those present in the GIT to form a complex that is poorly soluble or is unabsorbable. Thank you.